Hello, and thanks for joining our presentation today. The following is the second part in a series of webinars on bioavailability-enhancing technologies that we'll be presenting over the coming year. I'm David Vodak, Vice President of Bend Research, part of Capsugel's Dosage Form Solutions unit, and I'll be moderating these sessions. Today's webinar will cover important aspects of our spray-dried dispersion technology, specifically around performance, stability, and manufacturability. I'm joined today by Brett Caldwell and Dana Sattel, who will be helping present on this topic. Brett will be taking us through aspects of the performance of spray-dried dispersions, and Dana will talk to us about manufacturability of these formulations. And I'll do a brief overview of our understanding of the physical stability of these metastable formulations. Before I get into the technical portion of the presentation, I'd like to say a few words about Capsugel Dosage Form Solutions and Bend Research. Recently, Bend Research joined Capsugel's Dosage Form Solutions to bring two leaders together and enhance an offering for providing problem-solving in drug delivery solutions from preclinical to clinical and commercial. Bend Research is a leading scientific research, development, and manufacturing company, and as I said, now part of Capsugel's DFS. Bend Research's mission is to solve our partners and clients very hard problem statements in the guise of providing technology solutions and manufacturing formulations for delivering on programs from feasibility through clinical and commercial. We have about 250 employees with a very broad and diverse background to be brought to bear on these challenging problem statements. And our core values are listed here. The keys that I'll call out are the curiosity, science and engineering excellence, and the reliability and focus. We also have a strong intellectual property portfolio, and our business model is around providing that technology to our clients and partners, as well as developing new IP in the guise of technology development programs. Capsugel's DFS and Bend Research utilize an array of proprietary technologies and specialized manufacturing capabilities to solve customers' most pressing product development challenges, with a key focus on bioavailability enhancement. We also utilize full array of modified and targeted release technologies, as well as specialized encapsulation approaches for highly potent APIs and abuse deterrence. Additionally, we have expertise in biotherapeutic processing and inhalation formulations, leveraging Bend's particle engineering expertise and Capsugel's DPI capsule technology. The company's fast-to-clinic program streamlines product development from pre-formulation through clinical and commercial supply for finished dosage forms, where we choose the right dosage form for the product concept. We look at all these technologies, and we think of these as a toolkit where we made up the right problem statement with the right technology to advance the compound and ultimately get medicines to the market. So that'll wrap up the general short introduction, and we'll get into the technical portion of this presentation. I'll take a few minutes and introduce a kind of an overview summary of the spray-dried dispersion technology, and then I'll turn it over to Brett, who will take us through the performance and the key performance attributes of these formulations. I'll step back in and discuss briefly how we think about the physical stability of these metastable systems, and then turn it over to Dana, who will take us through important aspects of manufacturing and scale-up of spray-dried dispersions. So briefly, we'll just take a minute and talk about why we're even talking about spray-dried dispersions. And effectively, this technology is very important for compounds that have low solubility and or low permeability. And these compounds are considered the BCS2 or DCS 2A and 2B, where we look at dissolution rate limited for absorption and solubility limited for absorption. The spray-dried dispersions are most applicable for compounds that fall into this portion of the classification system, but can also be used for DCS4 compounds where enhancing the solubility can improve the permeability in as much as increasing the driving force. This is a slide that's a follow-on from the initial webinar where we were discussing how technologies are selected, and our approach is to think about what the compound properties are, what the product concept, the dose, dry powder, liquid, what the product concept is, and use that to select the technology. And this is a conceptual guidance map for when a particular technology is appropriate for a given compound based on, in this case, we're looking on the y-axis at aqueous solubility and the x-axis as log P. And so we can think about cases where if we have very high solubility and relatively low solubility, crystalline drug is probably going to be the right choice. As we decrease solubility, we go through a property space where just making higher surface area systems will be appropriate for getting the high level of bioavailability. And then as the compound solubility at a given dose decreases even further, we need to take extra measures and use either metastable SDD systems where we're kinetically trapping a higher energy form of the drug to enhance the solubility, or actually use lipids and the physiology associated with absorption of that to enhance the bioavailability. This slide is a snapshot, high-level snapshot of what we're talking about when we say spray-dried dispersion technology. So effectively, we take drug and a high glass transition temperature polymer and we dissolve it in a volatile organic solvent. That solvent stream is then pumped through an atomizer into a drying chamber where hot drying gas flashes the solvent off of the atomized droplet, creating a rapid drying kinetics and essentially freezing that solid solution into a dry powder particle state. And we'll talk more about exactly what that particle consists of in the coming slides. But essentially, we're looking for a homogeneous system where instead of having two amorphous glass transition temperatures, we now see a composite single TG associated with that homogeneous system. And in that case, we're looking for, does the formulation dissolve rapidly? Does it supersaturate relative to crystalline? And can we sustain that supersaturation? And then looking at how to put that drug form intermediate into the appropriate oral dosage form, be it a tablet, capsules, or even powder in a bottle, are appropriate. This slide is just meant to say amorphous dispersions have precedence and they are successful when you make them up against the right problem statement. I've listed a review here recently where the authors went into the literature and pulled out over 100 studies of different approaches for putting amorphous dispersions into in vivo models, and over 82% showed that this does work when that's the right problem statement. And you can probably suspect that the ones that didn't work, maybe they didn't have the problem statement exactly right. And then we have multiple products on the market, and we see this on an uprise in the coming years. We'll expect to see many more products that are launched using this technology. And then, as with the previous map, here we are looking at how we think about how to formulate amorphous solid dispersions. And in this case, what we're looking at on the y-axis is the driving force for crystallization, or the melting point to glass transition temperature ratio, plotted against the log P, or lithophilicity of the compound. And we have guidance maps like these to help us, before we start to make any powder at all, we look at our historical experience with this technology and the compound properties of what we're receiving, and say, last time we saw a molecule with these types of properties, what types of formulations worked, what were the problems we're trying to solve. And so using maps like this, we can capture that historical experience. And here we've listed some example dispersion polymers, and we usually break the world up into ionizable polymers and neutral polymers. And in the case of ionizable polymers, such as HPMCAS, these polymers have special properties in that when they're in the neutral state, they're effectively fairly hydrophobic. Therefore, they don't pick up a lot of water, and that leads to enhanced physical stability. However, when they wet in a physiologically relevant media, the succinate groups deprotonate, and what you end up with is a amphiphilic charged polymer molecule that can then have surface activity and effectively stabilize high energy forms of drug in colloids. From the neutral perspective, these are also very useful materials, and oftentimes we'll use neutral polymers when a drug has acid sensitivity or requires gastric dissolution for its specific absorption profile. And with that, the purpose of this webinar is to really look at the intersection of how each one of these performance, stability, and manufacturability come together to deliver a commercially relevant formulation. And the way we see these this Venn diagram interacting is, if you develop something that performs very well but you can't manufacture it or it's not stable, no one really cares. It's not a progressible formulation. And so it really is important, even at early stages, to think about how these circles come together and intersect to come up with a formulation that you have confidence is going to be robust for downstream processing. With that, Brett, I think I'll let you take over.
Thank you, David. This is Brett, and I'd like to provide a summary of how amorphous dispersions reproducibly increase absorption. That comes basically from coming up with a speciation model, or a mechanistic model for how SDDs work. We'll start with that. Understanding the mechanism leads to a need to identify tests that confirm you have the mechanism in place and in hand. You can use complementary and orthogonal tests to achieve that understanding. And then using those inputs with that understanding to develop biomodels that help you simplify your development pathway and limit the amount of in vivo testing that must be performed. So we'll start with a conceptual model of dispersion species. And this is a complex slide, but it can basically be distilled down to the statement, nothing about the performance is magic. It can be measured and understood and therefore utilized to your advantage during the development pathway. I'd like to start with the left side of it, and that's the goal of an IR SDD dosage form is essentially to release SDD powder as rapidly as possible, believing that powder and its characteristics are vital to the effect that we want to have in the body. The main point, though, in the key species are that the free and the micelle-based drug species that actually permeate the mucus layer, allowing free drug to permeate the lipid bilayer and into the plasma, those are the prime species of importance to absorption. Ionization can certainly matter for a drug form. It's not called out specifically in this particular diagram, but it is certainly tracked by in silico pharmacokinetic models. The drug nanostructures that come from SDDs, and we'll talk about in detail on the next slide, are key because they rapidly replenish these key free and micelle drug concentrations as absorption is occurring and pulling the dissolution concentration below sink. And finally, the kinetic rates and solubility values really combine to be critical for the overall performance. You're trying to avoid or retard the right side of this diagram where you have precipitation or conversion to a lower energy form. We'll call out in later slides how the composition and process of a dispersion can impact the appearance or rates of appearance for these particular entities. And the key for all of these tests then is to get the rates and extents of these important species and therefore use those to predict your performance in vivo. Dispersions generically work, but each drug interacts with the polymers and the physiology of the body differently. So in reality, we commonly use two models to frame our understanding of how dispersions are working, and the reality lies between these idealized situations I'll describe here. The primary difference in both of them is the concentration of observable nanoparticles, as you'll see as we go through this. So the first model, commonly if you're working with the dispersion, you believe that you have a low water solubility drug. And that brings the formulator to a dispersion, so we're really thinking about low solubility in water or in a hydrated polymer situation. So as we go across the top of this diagram, we're seeing a dry SDD particle hydrate and then form some nanoparticles. We think this in two different ways. Either it's association of the drug with a hydrophobic polymer patch, or that the hydrophobic drug is now being capped by some piece of the polymer. As those nanostructures are created and composed, the drug particle continues to swell and start to break up. It releases these nanostructures, which allows the free and the micelle drug concentrations to be rapidly replenished from these. So it is kind of driven from a phase separation of the drug with the hydrated polymer. And particle size doesn't tend to be a strong determinant of performance in this case because of the rapid nature of these interactions. The bottom part of the model is different, and it's usually, we see this with an ionized drug form or something that has a fairly water-soluble side of it, maybe an amphiphilic molecule. Usually these drugs have high solubility in the hydrated polymer, and therefore it acts more like a standard matrix tablet, where you'll get a lot of erosion and free and micelle drug leaving and being formed, and this particle only erodes at the rate at which that saturation in dissolution environments takes place. So we get diffusion of drug through this boundary layer driving dissolution, and when the drug is dissolving it, now you have a hydrophilic polymer and this particle continues to erode. So with that detailed description, we look at one major test protocol we use to characterize those species. Most of these tenets are very similar to literature tests, though. This is a non-sync microcentrifuge dissolution test. We commonly use biorelevant dissolution media, including transfers if necessary. Various mixing and separation techniques are out there, and this particular protocol allows us to use suspension or powder dosing. The key is to separate the three major species representing total dissolved drug from that precipitate or undissolved material. So back to the diagram, it's the free drug, the micelle-based drug, and the nanoparticle drug. What we're looking for is the rates and extent of these appearing or disappearing, and using that to get into the understanding that we have the mechanism right and how it will perform in vivo. The test rapidly helps to identify the technical challenge or the formulation challenge for any drug in this particular space. David showed the map before with the problem statements called out. Here what we're talking about are the specifics of that relation to performance. So for the top example, we're talking about drugs with a high propensity to crystallize. We called that out on the initial slide as the physical stability challenge, but it has performance ramifications also. The rate of precipitation is directly tied to the dissolution rate and the appearance or the concentration of a precipitation inhibiting polymer. So with a good SDD, we see a rapid dissolution rate and sustainment of that concentration. If we've had a drug that doesn't have the right polymer with it or too high of a loading, we will see this spike and crash related phenomenon that will leave us with concentrations by 90 minutes in the biorelevant regime that are similar to crystalline drug. In the alternate case study on the bottom, we're talking about drugs that don't have a strong propensity to crystallize, but they're very lipophilic and therefore hard to get dissolved. What we need here is a rapid dissolution rate, and particle size, as determined by the process, can strongly determine the success or failure of a dispersion in this regime. as David called out, there's many polymers that are used for dispersions. We commonly use HPMCS because it's a primary example of a precipitation inhibiting polymer. But the properties of HPMCS across compendial space, as shown by this whole graph, can vary widely. There's 3 nominal grades that are sold as identified by the L, M, and H boxes here. But compendial space actually allows a much wider understanding of how your drug interacts with that polymer. And almost in a QbD sense, you can tune your polymer tenets, whether that's performance or stability, based on using different polymers. For example, a model compound shown here with various substitution amounts of HPMCS, the microcentrifuge is shown in the top left. As we change the polymer substitution amounts and ratios, we see that the technical challenge of rapid dissolution with precipitation inhibition for this drug, we take the data from the left and translate it into the right, where now we're identifying that something with a succinate to acetate ratio of less than 1 is better for this drug than something with a more hydrophilic balance, something with a higher ratio. Across the bottom is a different drug, itraconazole, that has a different problem statement. It's in that high log P, needs to rapidly dissolve, and we use a more hydrophilic grade to optimize its performance. This is done in parallel with the physical stability characteristics, and you may end up later choosing a polymer based on a different, maybe stability ends up being a bigger challenge than performance. Finally, back to the theme that SDD performance can be measured and modeled, we like to use orthogonal or complementary tests to completely understand and validate our mechanistic beliefs. We called out drug and micelle-based concentrations as key, and I'll just summarize in the far right column that we can use a stronger centrifugation step to isolate the nanoparticles from the free and micelle drug. We can use NMR because nanoparticles are invisible to NMR detection, or we can use tests with permeability built in that only rely on the free drug concentrations. We can also use a function of dose or micelle concentration to build a holistic picture of the performance dependence and help predict in an in silico environment how these things will perform. Dosage form is a key piece of all performance manufacturer stability. Dosage dispersions can be used in a wide variety of dosage forms. This slide is monolithic based, but we also use multiple particulates with dispersions regularly, both for an IR or modified release. The usual goal for an IR SDD dosage form is to do no harm, essentially. We want to understand that we're releasing a high energy dosage form as we expected to from our powder testing. This just shows one example where we've created a tablet that disintegrates rapidly back to the primary particles using the same tenets of gastric to intestinal transfer and testing powder versus tablet to show that we get the same supersaturation, the same rates, and no precipitation. And finally, once you have the mechanistic understanding and the concentrations available, we translate these into biomodels, and the use of the biomodel changes throughout development time. You can use a problem statement like in the bottom left, where now that if you envisioned a solubilized form, you can choose a drug that binds better than something that was previously soluble but didn't bind as well. Or in a more life cycle management or late stage dispersion program, you can actually usually get, as we'll talk about later, you can attach the process parameters, for example, all the way to performance and understand how process changes may affect your bioavailability.
Okay, thanks, Brett. That's very informative. So now I'll step in and say a few words about the physical stability of these metastable systems. So to start with, it's important to think about, where are we at with this the spray dried dispersion? What's the initial SDD state, and what's the final state at infinite time? And so, essentially, there's two ways to think about it. In the first case, the top case, what we're looking at is a solid solution. Effectively, the drug is dissolved into the polymer. It's a thermodynamic system, and effectively, if you let that material sit over an infinite amount of time, it will never come out of that solid solution. It will always be homogeneous. The limitation with that is oftentimes drugs don't have very high solubility in the polymers, and therefore you are limited in your loading. Another challenge with that type of a formulation is that when you're talking about thermodynamics, that is at a given temperature and given condition. And that phase diagram is going to change as you change temperature or storage condition. And so it can oftentimes be challenging to be predictive about physical stability in those cases. The place where we generally live in the spray dry dispersion world is a place where we depend on kinetic stability. And in those cases, we start from a homogeneous SDD, and over an infinite amount of time, it will eventually phase separate and either phase separate into amorphous systems or crystalline. And as I'll get into on the coming slides, it's important to note that we are in a kinetically stabilized regime, which allows us to do some things in terms of accelerated storage and predictive modeling around the stability of those systems. So if we take a very simplistic view of what I attempted to describe on the previous slide, here we're looking at some Flory-Huggins theory, and as relates, we're looking at the free energy of mixing as a function of drug loading into the polymer. And from freshman chemistry, remember that if the free energy is negative for a system, then it spontaneously occurs. And so everywhere where the line is below zero, that means that the material is effectively soluble. But as we swing up the slope there in the metastable regime, now it's not necessarily a two-phase, but because we've pre-mixed it, that material is going to stay in solution. However, if we give it mobility, it will relax and phase separate out. And that's an important thing to note. So once we've crossed the bridge that we're going to be in a kinetically stabilized or metastable state, it's very important to note that we start with a homogeneous SDD, and that ties back to the process, which Dana will fill in as I wrap up my description of physical stability. But here we are looking at a ternary phase diagram where we start with an initial spray solution composition where we're largely solvent. And as we do the spray drying process, we're going to be removing that solvent. And on the phase diagram, that means that compositionally we're going to be going through some very important phase boundaries. The first of which being the polymer gel point, and the second being the solubility of the API in the solvent that you're spraying out of. So compositionally, as we spray dry, we cross both of those boundaries. And it's important that if at all possible, we try to cross the polymer gel point before we are supersaturating the API in the spray solution. And that will do is it will limit the chances that we're going to have drug precipitating out in the droplet, which would lead to a heterogeneous SDD, and then make it very challenging to stabilize these or even be predictive about what the physical stability is going to be. Our approach to physical stability is very much a phase appropriate, where early on we will use some simple experimental tests to tell us whether or not we're going to have a problem. And then as the drug advances and we start to know more about it and the program is advancing, we'll get into some more sophisticated experimental technique and model to effectively show that we know the system very well and that we're confident at a given storage condition we're going to have greater than 2 years stability. And then finally, unfortunately at this point, with filing and regulatory, despite having the models around knowing that it's going to be stable, we still have to collect the data and show that our models are right. So early on in the screening process, first we'll go into, here's just a general, I'm not going to go through all the text here, but here's just a general description of some of the analytical tools that we'll use for monitoring physical state or stability. Some of the examples like scanning electron microscopy, that's a very qualitative technique, but it's probably the most sensitive technique on this list in that typically crystals will form on the surface of SDD particles because they don't have to suffer the energy penalty of pushing a high TG polymer out of the way to grow. We also do a lot of modulated DSC, again looking for phase separation. And then, as I'll talk to later, thermal activity monitor, isothermal calorimetry. So with modulated DSC, some of the first tests we'll do with formulations is evaluate the glass transition temperature of a dispersion as a function of relative humidity. And we'll relate this to potential storage conditions that the sample might see, and then use that as a guide to say, do we expect we're going to have an issue with this dispersion at some of the relevant ICH storage conditions. So in this plot, we're looking at the glass transition temperature on the y-axis, and on the x-axis is the relative humidity at which it's stored. So you can see how as water plasticizes the dispersion, and we increase the RH, the TG of the dispersion is going down. And what we're showing here is how it relates to different ICH stability conditions. And what we look at is that delta between the storage condition and the glass transition temperature, and use that to guide whether or not we think a formulation has the potential to have some physical stability issues at a given condition. So in this example, what we're showing is that when the TG is 40 degrees higher than the storage condition, here we show with this SEM, no crystals are growing anywhere after 18 months of that condition. Whereas when the TG is less than 10 degrees from the storage condition, even after 3 months, we're starting to see crystal growth. So that's the way that you can use this type of analysis as a guide. And if we take a look at our kind of historical knowledge of using this kind of rule of thumb, here shown in this histogram, on the y-axis we have fraction of compounds that are stable after a 6 to 13 week stability challenge as a function of the delta between the TG and the storage condition. And you can kind of see that anywhere above kind of 20 degrees C difference, we rarely ever see an issue, and probably in those cases where we did, I might challenge that we didn't start with a homogeneous SDD. But as you get closer to the 20 to 10 to 0 delta between storage condition and TG, we start to expect to see issues. And the other thing you'll note on this histogram are the numbers in each of the bar plots. And those numbers are talking about the number of compounds that are in each of those data sets. And so you can also see that, we just generally don't formulate. We try very hard not to formulate such that we're going to expect to have a TG 10 to 20 degrees above the storage condition. So this is a really simple way of, with a small amount of material, coming in and saying, yep, physical stability looks like it's going to be straightforward, or we may want to consider packaging. As we advance a compound along, we'll get into some more sophisticated physical stability mapping. And in this case, on this slide, we're describing using isothermal calorimetry to actually measure crystallization kinetics. And so schematically here, we're going to put the samples up at accelerated conditions where they're at, or even where the temperature's at or even above the glass transition temperature of your dispersion, and then watch the heat flow as these systems phase separate and then crystallize out. And the cartoon here kind of describes how that crystallization exotherm is then translated into the integral of that. And what we'll do with this in terms of modeling is we'll look at the integration of that crystallization heat flow signal, and just kind of based on what we can typically robustly quantify in, say, a powder X-ray experiment is kind of 5% crystallinity. So we'll map out the time it takes to generate 5% crystalline material, and then plot that against different storage conditions to generate a physical stability map. So on this slide, we're showing that in the bottom plot on the bottom left. We're looking at the log of the time to 5% crystallization versus the TG over T, Kelvin to Kelvin. And effectively, we'll set up samples at conditions higher than the TG of the dispersion and watch them crystallize, and then we can put those on this plot and then extrapolate out to 2-year stability, so that we can say at a TG over T condition, in this case of about 1.05, we expect to have 2-year stability, and then we can go back to our storage temperature versus relative humidity map and generate plots where the Tg over T is always 1.05, and therefore generate these iso lines and use this to guide our stability packaging. This slide just shows that in addition to all the models that we use to be relatively sure that we're going to observe physical stability, when you put these up and generate the actual data, we in fact do have long-term physical stability. And so with that, I'd like to turn it over to Dana to take us through the manufacturing portion of the talk. Thanks, Dana.
Thanks, David and Brett. We'll end the webinar here with a discussion of SDD manufacturing, including an overview of the spray drying equipment and process, a methodology for defining key process parameters, and some considerations for spray drying scale-up. On this slide, we have a general layout of a typical spray drying equipment train for SDD manufacture. We begin by dissolving a drug and a polymer in a solvent or a solvent blend to make a spray solution. That feed solution is then pumped to a nozzle where it is atomized into droplets. Those droplets are contacted concurrently with a hot drying gas stream, usually nitrogen due to the flammable nature of the solvents and dust that we work with. That drying gas is also introduced at the top of the spray dryer. The droplets dry to solid particles inside the drying chamber and are pneumatically conveyed to a cyclone separator, where the majority, usually about greater than 90% of the SDD product, is removed from the gas stream using the centrifugal forces generated inside the cyclone. The remaining solvent-laden drying gas is passed through a police filter to remove fines, and then in larger scale spray dryers it's recycled through a condenser to remove the solvent vapor, and then back into the drying gas heater and into the drying chamber. Smaller scale equipment is often operated in single pass or open loop mode, and in those cases a fresh nitrogen gas stream passes only once through the dryer and is vented to a waste stack. Since the residence times inside the spray dryer are short, on the order of seconds up to maybe a minute at most, the SDD that comes out of the cyclone will have some residual solvent remaining on the particles. That residual solvent is removed in a secondary drying process, usually a tray dryer or an agitated conical vacuum dryer, with longer residence times that are more on the order of hours, in order to remove that residual solvent down to acceptable levels. The spray drying process has some key aspects that make it ideal for making amorphous solid dispersions. If we review the key physical situation, we see that the key control volume is up near the nozzle, where the droplets are formed by the atomizer and immediately contacted with hot drying gas. The drying happens very quickly, on the order of milliseconds. The skin forms and eventually further dries to a solid particle. This rapid drying or quench rate can enable kinetically stable dispersions above the thermodynamic solubility of the API in the polymer. So as soon as the droplets are formed, the solvent begins to evaporate, cooling the droplets to the dew point of the solvent. As the particles dry, they warm to a temperature that is equivalent to the spray drying outlet temperature as a maximum, making the spray drying process ideal for heat-sensitive actives as well. We're showing some computational fluid dynamics modeling outputs in the lower right-hand corner. One output is a temperature contour showing the majority of the drying chamber is at a mild temperature, which is equivalent to the outlet temperature. And the other output is a particle trace showing wet droplets in light blue, skinned particles in red, and dried SDD particles in blue, demonstrating just the fast drying kinetics of this process. Another key aspect of spray drying is that the SDD attributes such as particle size and density are easily tuned to a specific target or optimized by selecting the appropriate droplet formation and droplet drying parameters, and we'll talk more about those on the next few slides. And lastly, the spray drying process is commercially demonstrated. It's been around for hundreds of years in the food and chemical industries, and it's easily scaled from milligram quantity up through metric ton quantities. On this slide, we're showing a methodology we developed for the efficient identification of spray drying process parameters after working on numerous SDD processes. The purpose of this methodology is to reduce the development time and the number of spray drying development runs or API demand required to identify an optimal process that yields the SDD with the target quality attributes. To do this, we take advantage of models and tools to identify the spray drying parameters in silico or with data from the offline tools prior to doing any spray drying development work. The knowledge generated by using this methodology, since it is highly model-based, also provides a good foundation for quality by design efforts. We won't go through each box today, but we've provided a reference there where you can read a little bit more about this methodology. There are two key physical aspects of the drying process and spray drying parameter sets, if you will. The droplet formation or atomization parameters and the droplet drying or thermodynamic parameters. We'll go over both of these and some of the models and tools to define these in the next few slides. So this graph shows a thermodynamic operating space based on simple mass and energy balance equations that are summarized there on the right-hand side of the slide. On the x-axis is the drying gas inlet temperature, and on the y-axis is a ratio of the spray solution feed rate to the drying gas flow rate. And we use this ratio that we've defined as a specific drying ratio so that we can plot multiple spray drying scales on the same plot. But since drying gas flow rate is essentially held constant at each scale, you can think of this y-axis as a spray solution feed rate. And as you go up on this axis, spray solution feed rate or dryer throughput goes up. So within the graph are contours of constant dryer outlet temperature and isolines of constant relative saturation at the dryer outlet. The lower right-hand corner is the hotter, drier space, and the upper left-hand corner is the colder, wetter space. This type of model can be used, or in graph, can be used to lay out your operating space and identify your key thermodynamic parameters. For instance, a maximum inlet temperature can be determined based on either properties of the material, say its browning temperature, which you may want to stay below to ensure that if there are any deposits near the lid, which can have local hot spots so that they don't degrade or brown, or there may be just equipment constraints with, say, the process heaters limiting your maximum inlet temperature. You can limit the relative solvent saturation. You want to stay below a value such that the dryer is low, the relative saturation within the dryer is low enough to prevent spray drying. And this can be assessed by evaluating the TG of the SDD at various solvent saturations. In order to achieve an efficient process, there's likely a minimum spray solution feed rate that you can define. And last, you can define a maximum or target outlet temperature based upon SDD product attributes or thermal stability. And we'll talk more about that aspect, correlating the outlet temperature to the SDD attributes in the next slide. Okay, the SDD product attributes like density or compressibility often correlate well to the outlet temperature and relative saturation at the dryer outlet. Due to the nature of the film-forming polymers used in SDD manufacture, we see a skin formation on the droplets immediately after the solvent evaporation begins. If the drying rate is slow, or if the spray drying parameters are cold and wet, this skin remains pliable as the particle forms, and the resulting SDD has a shriveled raisin morphology with high density and low compressibility. If the drying rate is fast or hot, dry spray drying parameters, this skin is more rigid and the solvent diffusion through the solidified skin is slow. The partial pressure of the solvent can build up inside that droplet skin, resulting in an SDD with a more balloon-like morphology that has lower density and higher compressibility. So we'll show some videos of droplets suspended on a thermocouple and subjected to first cold, wet drying conditions. Demonstrating the raisin morphology, and second, the hot, dry spray drying conditions illustrating the faster droplet drying and the balloon-like morphology. So switching now to the droplet formation or atomization parameters, a pressure swirl nozzle is shown on this slide. Well, there are many different types of atomizers that can be used in the spray drying process, tube fluid, rotary, all different types. The pressure swirl nozzle is preferred for SDD manufacture just due to its ease of use and robustness of operation. It also generates really pretty narrow droplet size distributions and is readily scalable. The spray solution is fed at a high pressure through the swirl channels, which are designed to impart an angular momentum to the fluid, causing it to fan out as a liquid sheet on the inside of that orifice chamber, and then exit the orifice as a swirling liquid sheet or hollow cone. The liquid sheet thins out to ligaments and then breaks up into droplets. The droplet size distribution produced by the nozzle is a function of nozzle geometry, things like the swirl chamber design, an open area, the orifice size, and even the surface finish of the orifice and swirl chamber. And also the nozzle operating parameters like nozzle pressure and feed rate. And then lastly, the solution properties, viscosity, density, and surface tension. Since the physics of ligament breakup into droplets is influenced by things like turbulence and instability, it can be difficult to model from first principles. But there are quite a few literature examples, entire books written about with these types of correlations. And we've shown one on the right here that we found, right-hand side of the slide, that we found to be particularly applicable to SDD droplet size predictions. And these correlations take into account the three main buckets of geometry, operating parameters, and spray solution parameters. Since the droplet size can be difficult to predict from first principles, we measure it directly by a phase Doppler particle size analyzer. The PDPA measures droplet size distributions, velocities, and fluxes, and we can often use placebo spray drying solutions, so no active may be required. We can use this data to assess the nozzle performance, select a target nozzle geometry and operating parameter before we do any spray drying work. The data is also used to build the empirical correlations like the one we showed on the previous slide, and for predicting droplet size from those empirical correlations. And we also use the nozzle test stand just to assess nozzle-to-nozzle variability that might be present from manufacturing or assembly tolerances. So in summary, this chart shows the key spray drying process parameters grouped by those aspects of thermodynamics or droplet drying and atomization or droplet formation. The top row, I won't go through every box, but the top row lists the key independent parameters and briefly summarizes the selection criteria and constraints. And the second row shows the reduced set of dependent parameters like outlet temperature and relative saturation that are often highly correlated to the SDD attributes like density, residual solvent, physical state, and particle size. So moving on to the discussion of scale-up, we have various spray drying equipment scales available here at Ben Research, and they're shown on this slide. We have several lab-scale spray dryers for early screening and preclinical process development, several high-efficiency clinical-scale mobile miners in both our development labs and our GMP facility, and a larger clinical and commercial-scale high-efficiency PSD2 in our GMP facility. We're also in the middle of installing additional PSD2 equipment in both our development and GMP labs. So beyond the PSD2, we also have experience defining processes and scaling up to PSD3, PSD4, and PSD5 scale spray dryers. So by matching the droplet size and the drying rate or thermodynamics across each scale, the spray drying process can be easily scaled up from screening milligram quantities up to metric tons. There are two main considerations for matching the droplet drying rate or thermodynamics. One is the whole open loop. In smaller units, the drying gas is not commonly recycled, and the smaller dryers often operate in open loop, while in larger dryers, the drying gas is recycled through the condenser and back into the drying chamber. So this recycled solvent vapor must be taken into account, since it will contain a significant amount of vapor that will impact the thermodynamics and the drying rate. So in order to predict this effect and account for it early in development, we often operate our smaller scale spray dryers in closed loop mode to mimic the larger scale spray dryers. And we'll talk about the effect of condenser temperature on the thermodynamic operating space in the next couple of slides. But the second aspect to think about for matching drying rate or thermodynamics is the density of the spray drying plume, or the droplet density of the spray drying plume, which increases proportional to the feed rate across scales. And there are just many more droplets in nearly the same key control volume size. And this increased droplet density can cause a resistance to efficient heat transfer from the drying gas into the plume and mass transfer of the solvent vapor out of the plume at larger scales, effectively slowing down the droplet drying rate at larger scales. However, this effect can be easily mitigated, predicted with scale factors, and mitigated by increasing the drying temperatures at larger scales. So on the atomization front, in order to match droplet sizes across scales, higher nozzle pressures are required. And again, this should be evaluated early in development to ensure that a target SDD particle size is selected that can be achieved on larger scale spray dryers. So we use the nozzle test stand to help us do that. And we're showing here a graph of PSD1, PSD2, and PSD4 scale pressure nozzle data from the nozzle test stand, droplet size and solution flow rate versus nozzle pressure. So in order to achieve that same target droplet size of 70 microns across a 9x solution feed rate, the pressure is increased from around 300 psi on the PSD1 scale to greater than 1200 psi on the PSD4 scale. So you can see why it's important to keep this in mind early so that a reasonable target SDD particle size can be selected early in development. So switching back to thermodynamic parameters, this is the same graphical representation of the mass and energy balance or thermo model that we showed previously. In this case, we've added a third axis for the condenser temperature, which is the additional key parameter that must be considered at large scales. So again, we're showing dryer inlet temperature on the x-axis, condenser temperature on the y-axis, and the specific drying ratio or feed rate on the z-axis with contours of outlet temperature within the box. We can also plot contours of constant relative saturation within the space. So in this type of 3D box, the colder, wetter spray drying conditions are shown towards the back left-hand side, and the hotter, drier conditions are shown towards the front corner. So again, similar to the thermodynamic operating space we reviewed in 2 dimensions earlier without the condenser temperature, we can lay out an operating space or map out an operating space. We've plotted the experimental conditions for a 25% HPMCAS SDD at PSD1, PSD2, and PSD5 scales within this three-dimensional operating space. In this example, we show equipment constraints on maximum inlet temperature, that's the red plane, and equipment constraints on minimum condenser temperature, that's the blue plane. Both of these constraints are based on plant utilities and actual equipment capabilities. An operating constraint at 40% relative saturation is shown with the green plane. So beyond that plane, the drying process is just too wet and there isn't enough driving force for drying, leading to spray painting and low yield. So you can see that there are no actual experimental runs beyond that because the yield starts to drop off in the back right-hand corner of the three-dimensional space. The SDD attributes like compressibility or density or residual solvent can be predicted for every combination of parameters within that space, and a plane of, say, target SDD compressibility that is shown here in pink can be defined. So every place to the right of that pink plane has acceptable SDD compressibility. So the design space or operating space, if you will, is shown on the insides of all of those planes in the three-dimensional space. So earlier we showed that SDD density or compressibility often correlates well with outlet temperature. The added condenser temperature parameter, and to better account for scale, we developed a dimensionless parameter that takes into account all of the key thermodynamic process parameters. A dimensional analysis was performed on an equation for the rate of heat transfer into the droplet and the rate of mass transfer out of the droplet. And that resulting dimensionless parameter was evaluated using the easily measured and available thermodynamic process parameters like temperatures and flows, and correlated to the SDD compressibility as measured by mercury intrusion. So in the interest of time, we won't spend much time reviewing the derivation of the HMT function or the mercury intrusion test for compressibility, but I just want to briefly introduce the topic here. So this graph shows the example correlation of HMT ratio to SDD compressibility for a scale-up of 25% of a 25% HPMCAF SDD from the PSD1 scale to the PSD5 scale. And this is a really big scale-up, actually going from 20 kilos per hour spray solution up to 500 kilos per hour spray solution. So about the biggest scale change you can see. As the HMT ratio plotted on the x-axis increases, the rate of heat transfer into the droplets is faster than the rate of mass transfer out of the droplets, and the SDD density increases. So to identify process parameters to achieve the target normalized SDD compressibility ratio of 1, which in this case was ideal for this particular SDD in terms of its downstream roller compaction process. You can use at any scale, you can use the HMT correlation. So for example, for a PSD5 dryer, at a target HMT ratio of around 0.8 is desired, and that HMT ratio is calculated using the key thermoparameters that we reviewed today. The HMT equation is shown on the right-hand corner, and you can see that it contains inlet and outlet temperatures and relative saturation parameters that are calculated using the temperatures and flow rates that we reviewed earlier. So if the target HMT ratio is known, the target spray drying thermoparameters are also known. We can also use this correlation for linking back to things like SDD residual solvent or physical state. So you'll notice that there's also a scale factor. We won't cover that today, but that scale factor can also be predicted. So in summary, all of the work that we've described today forms the foundation of a quality by design effort. So it's all aimed at identifying the functional relationships or models between the target product profile and the SDD critical to quality attributes and the SDD CQAs and the SDD or spray drying process parameters. So I've listed some common SDD CQAs and process parameters here. We use different skill sets or tools along the bottom of the page to build these functional relationships or models that we've discussed today. So for example, under the sensors and analytical tool set, the dissolution work that Brett described is used to identify SC species and build mechanistic performance models and biomodels to predict in vivo performance. And the DSC and TAM work that David described earlier is used to build phase diagrams that predict physical stability or the state of the physical state of the SDD. So the science of scale experiments are used to build understanding of key material properties or process parameters. The example here was the novel test stand to identify atomization parameters. We didn't discuss risk assessments today, but those are also critical for identifying critical to quality attributes, and we use them throughout the development process. On the right-hand side, the experimental work that we highlighted and the modeling tools we described today can be used to build process models that identify a spray drying design space. An example of which is shown on the last slide here, just showing some aspects of what an SDD QbD package might contain. We have thermal models and atomization models to define your operating space and guide experimental work from a more fundamental or first principles perspective. Models to link process parameters to SDD attributes like the outlet temperature and HMT correlations that we reviewed to predict SDD density. Models linking SDD attributes to dosage form manufacturability like compressibility to predict critical to quality tablet attributes based upon the SDD attributes. and models to link SDD attributes to performance and stability, like the biomodels that Brett described to predict in vivo behavior. And all of the models constructed are based on the aspects of performance, manufacture, and stability that we reviewed today.
Great, thanks, Dana. That's very interesting, and you can tell we'll be able to go into much more detail potentially on a follow-on webinar. I'd like to thank everybody for joining, and just so we're aware, we're going past the hour here, but I think there's a few questions that we'll try to answer live here, and then questions that we're not able to answer, we'll follow up offline, either through email or phone calls. So please contact us with questions. For the first question, it looks like we have a question from around the process, and it says, droplet size seems to become larger as you move from PSD1 to PSD2.3 to PSD4.5 in slide 46. Can you actually match droplet size as you scale up by changing the gas pressure? Dana, do you want to try to field that one?
Sure. Yeah, that's a great question, and absolutely we can match droplet sizes across scale. I think in slide 46 we're showing pressure nozzles, so it's pretty, while it becomes more difficult to match smaller droplet sizes because you have to increase the pressure, it is definitely doable. And something that we've demonstrated quite a number of times as we scale up from PSD1 to even to the PSD5. So I think the important thing to keep in mind is to keep a reasonable target particle size early in development so that it's not too small, and then therefore the pressures become too high in order to achieve that. So.
Great, thanks, Dana. And a follow-on question that looks more around formulation is, how easy is it to make SDD in capsules for phase 1? Brett, do you want to try and tackle that one?
I will. This can be very easy, but it is not 100% generic. It gets back to determining the technical challenge for each dispersion or its critical quality attributes if you're thinking in a quality by design space. In our experience, non-sync tests of dosage forms as a function of capsule contents can reveal whether that will be easy or hard for your dispersion. And it commonly relates to the polymer selection or the drug properties. If it's very lipophilic, it can make things better or worse depending on the interactions with the polymer. So in many cases, whenever we have difficulty with that, we can formulate the powder or granules to improve the SDD and capsule performance.
Great, thanks, Brett. This looks like an interesting question. What is the typical API demand for early, i.e. pre-POC product process development and for a full factorial QbD commercial product development optimization?
Yeah, I'll take that one. I think this is a great question, also a little bit difficult to answer just because it depends upon the specific SDD and that SDD's critical to quality attributes. You know, does it have many critical to quality attributes or just a couple that are coming into play? Also, things that matter are, can we use a placebo for some of the development and, say, novel test stand work that we demonstrated in this webinar. So oftentimes if you can use a placebo and then rely heavily on sort of the models that we've demonstrated or discussed in this webinar, you can be very efficient, especially in the pre-POC. And then when you get to the QbD and the full factorial designs, you can design those, you know, sort of from first principles, again, in a really efficient manner. So don't have an exact number for you, but those are some of the thoughts that come into play.
Great, and again, that seems like something we could go into more detail, Dana, exactly, in maybe a follow-on webinar specific to the manufacturing. It looks like we have time for a few more questions, so this looks interesting. Will spray drying technology assist the future of solid SED/SNEDs in the days to come, and is Ben planning to grow in this direction?
Getting back to the compound space that we probed, the earlier maps where there was a propensity to crystallize on the y-axis and log P or lipophilicity on the x-axis. The bottom right of that graph is the space where solid SEDs and SNEDs would be the space where you'd formulate those types of drugs. Commonly the problem statement there is dissolution rate, and you can certainly suspend or spray dry to put those types of drugs onto solid high surface area materials, and then those powders are easily handled for normal capsule or tablet dosage form development. We also can formulate those types of drugs into multi-particulates. We have a couple of processes and technologies that we use to make solid lipid pellets to formulate for pediatric or geriatric use, immediate or modified release, or coating them for taste masking applications. So certainly spray drying is one tool that we would use depending on the actual problem statement of the client.
So it sounds like absolutely we're in and around that area, and spray drying is a process that's going to play a big role. So it looks like there's a question here about how cyclodextrin is used to increase solubility. I don't know, Brett, do you want to try to field that one as maybe our final question?
We certainly have some experience spray drying cyclodextrins with specific drugs and applications. And commonly what you're looking for there is to just isolate a drug either in the amorphous state, and cyclodextrin is a very water-soluble entity to help with dissolution rate. Occasionally the drugs will go into the cyclodextrin cavities and therefore be protected in some sense, which helps increase the drug loading and decrease the propensity to crystallize for that drug. Cyclodextrins have been successful in certain projects. Usually the polymers that we discussed in the early slides are amenable also, but cyclodextrins can be spray dried and used for injectable applications and things like that on occasion.
Okay, great. I think that we've kind of gone over here. There are quite a number more questions, and we'll follow up with those directly. And if people that have dialed in have other questions, please contact us at the email shown here on the slide. As mentioned at the beginning of the webinar, we do plan to host additional webinars in the following year around bioavailability-enhancement, and hope that you will continue to participate. The next webinar in the series will address nanotechnologies, so please check our website to register. On behalf of Capsugel DFS, myself, Brett, and Dana, I'd like to thank you for joining us today. There's a survey that would please fill. There's a survey at the end, and if you could please fill that out, that'll pop up on your screen. We can use your feedback to improve these. So thanks again for your time and attendance today, and look forward to our next presentation. Thanks.